Hey, good afternoon, everyone, and welcome to our third Thursday Science Webinar series. My name is Bert Alexander. I'm a Science Officer at the European Marine Board, and I'll be the moderator for today's session. So, hold on. There are a couple of housekeeping rules for the session today. So it will be an interactive session. We'll start off with a half an hour talk, and then there'll be a question and answer session. So you can enter your questions in the Q&A box and let us know which organization you're from, and also make sure that your name is clearly entered on Zoom so that we know who you are and where you're from. And then I will be going through the Q&A box to select the questions to ask our speaker. So you can also use the chat for any technical support questions. And the webinar is being recorded, and it will be available afterwards on our YouTube channel. So the topic of the webinar today is machine learning to unlock the potential of plankton imaging big data. So this is based on the European Marine Board Big Data in Marine Science Future Science Brief that was published in 2020. And we have with us here today one of the authors of the document and member of the working group, which ran from 2019 to 2020. So Dr. Jean-Olivier Irisson, and he is Associate Professor at the Sorbonne Université in France and part of the computational plankton ecology team of the Oceanography Lab of Villefranche. So over to you, Jean-Olivier. We're very interested to hear about the work that you've been up to over the past couple of years.
Thank you, Britt. So I'm going to share my screen, which you should see now. What? Okay, so you presented the science brief. I just added this slide to remind that, yes, this was in this context that I was invited to give this that webinar. And so I'm very pleased to be here today to talk to you about the use of machine learning to deal with the big data that is generated in plankton ecology by plankton imaging instruments. My goal is to show you how machine learning is used to go from pictures to knowledge, hopefully. And that's actually two big chunks: how we extract data from the pictures and how we then process that data to get to some results, basically. I'm presenting today, but this is the work of many colleagues in the lab and internationally. So the first question is, why do we study plankton? Why do we spend all this effort to collect that much data about plankton? And there's several roles that the plankton plays in the ocean. One big one is that phytoplankton, so the algal plankton, produces oxygen and is responsible for roughly half of the primary production currently on the planet. The life that takes place at the surface of the ocean gives rise to an export of carbon towards the deep sea, and that carbon is sequestered if it goes deep enough for centuries to millennia, and that contributes to the regulation of climate. And finally, almost all fish depend on plankton. So of course there's planktivorous fish when they are adults, but all fish go through a larval stage, at which point they eat plankton. And so the dynamics of plankton will influence the dynamics of fisheries too. So to study plankton, traditional approaches are to just deploy nets with fine mesh, or to get water samples. And after that, we saw the emergence, let's say, of several types of imaging instruments. So some imaging instruments, like the ZooScan that is on the bottom left here, are imaging instruments that do not change the way that the plankton is collected. It just changes the way that it's analyzed. Instead of looking at the result of the net catch under a microscope, we scan it and we take an image of everything, and this is the analysis. So that's the case of many instruments, but there's also other instruments, like a few that are presented here, that take the images directly in situ, so there's no sampling anymore, in the sense of there's a digital sampling, there's no physical sample. These in situ instruments, they give access to additional information in the sense that they are better resolved vertically, and we see the organisms in their original state in the ocean. So to give you an idea about the amount of data that these instruments produce, so you have the ZooScan, the first one that I spoke about. These are the type of images that it produces. And one ZooScan will produce about 1 billion pixels per year. But the important aspect is that, the important quantity is the number of objects, so number of, organisms like this, but also other objects that it will produce. And that is roughly 1.5 million per year in normal operation. So these 1.5 million objects, then they need to be identified taxonomically, data needs to be extracted from them, such as, they need to be measured. And this is the data that we'll be looking at afterwards. And so one ZooScan will produce 1.5 million of those, but there's hundreds of ZooScans throughout the world, so that quickly gives rise to big numbers. The UVP5, which is presented next, is an order of magnitude more, so about 10 million objects per year. And the in situ ichthyoplankton imaging system, or ISIS, which is the last one, is another order of magnitude that would be 100 million objects for a single cruise at sea. So in the end, what we have is complex data, because it's images which have this spatial structure, there's potentially rich information. In that case, it's only gray level, but there's also color ones, and there's lots of it. So another way to see the quantity is to look at the accumulation of the number of objects, the number of images in one database, where you see that since 10 years it has grown at a pace that is very rapid. And spatially, that means that we have actually quite good coverage of the world's ocean through these imaging instruments. The question then becomes, how can we actually deal with that amount of data in an efficient way to get to ecological conclusions, because that's the goal that we start with. And so that's the first step, which is how do we go from the pictures to numbers? Basically, in ecology, we want to have tables with counts of organisms per location. That's what we work on. Currently, we have a bunch of images for each location. So this is a process that is commonly called quantitative imaging, meaning that the instrument, they take images, but they take images in a controlled manner that we can exploit quantitatively afterwards, and from which basically we can compute concentrations. So we start from such an image, which is a ZooScan image, okay? And the first step would be to detect and extract all the organisms of interest. That's called segmentation, and that is automated. In the case of plankton images, this is actually often a simple step because the background is controlled or relatively simple. And so the segmentation is basically take everything that is darker in that case than a certain threshold, and we get the image, okay? Then the second step is to classify them. So one possibility would be to just classify them manually, but given the amounts of images that I told you about on the previous slides, you can imagine that it would be really cumbersome to do it this way. So a traditional way that this is done is through machine learning, combined with the intervention of human taxonomists to do this sorting. So there's two ways that this is classically done. Let's say there's the old way and the new way. The old way was measure plus classify approach. So that means that for each image that was segmented before, we measure stuff like the area, the length, we fit an ellipse on it, and we extract what's called features from those. that are basically numbers that summarize the content of the image. And then we have a table of numbers, and we can use a machine learning based classifier to classify the images. Just use something off the shelf. The one that is, there's several ones that are commonly used. So the idea is that you just train a classification algorithm with a few examples that are labeled. It recognizes that a certain type of plankton is always large, is always dark, and then classify the subsequent ones the same way. The new way is through what's called deep learning, and in particular convolutional neural networks, which is what is represented here. And basically a convolutional neural network does the two steps that we have seen here, but in just one model, but actually in two different parts. The first part, the original image is transformed, then summarized, and retransformed and re-summarized, and that gives a vector of features. These features, again, they are numbers that summarize the content of the original image. So they play exactly the same role as the classic features that I spoke about before, except they are extracted in a non-deterministic way, and they are optimized together with the classification process. And then after that, we have a classifier, which in that case is a multi-layer perceptron, and that gives the final classification as a taxon. So this approach of plankton image classification is actually a quite active field of research with 175 published papers until 2020. Currently the trend is continuing, meaning there's something like 20 papers dealing with that published every year. It has been a really active topic of research, including in the computer science community since competition on the Kaggle website in 2015. So Kaggle is a website for machine learning competitions, and one of those competitions was about plankton, which put it under the radar basically of people that are not really used to dealing with this, and that has motivated quite a few people to work on this and given rise to more paper and more approaches. In terms of approaches, actually, historically, there's been a shift in the different methods. The initial ones were artificial neural networks that are not convolutional, discriminant analysis, nearest neighbor, and then as soon actually as the algorithm of support vector machine and random forest appeared in the general computer science literature, they were taken up by the plankton imaging community and used in quite a few papers. And recently, since 2015, almost, these convolutional neural networks have taken over these previous approaches. So the question is why, why is it so difficult that it's not a solved problem after so much research? Why is plankton classification hard? Well, one reason that it is particularly hard as an image classification a challenge basically, is the distribution of images within the different classes that we're trying to classify. And this is the plot that is represented here. So that's the number of images per class. You probably cannot read the classes, but that's not very important. What is important is the aspect of that plot. Ideally, for a classifier, a machine learning based classifier, you want this to be flat. You want to have as many examples for each class. And what you can see is that it's far from being flat. There's a few classes that dominate largely all the rest. And then there's many classes that are very rare and therefore have very, very few examples. So that would skew a machine learning classifier towards predicting that everything is the big class and they will completely forget about the small ones. Oftentimes, the rare species that are indicative of something in the ecosystem, they are interesting, basically. So we want to get the rare ones too. Another problem is that if we look at this big class, which is often a class of detritus, so stuff that is not a living organism in the sea, so it's all classified together. They are not, sometimes not the main focus of the study, but they dominate, particularly for in situ imaging instruments that see a lot of marine snow. And these detritus, they can have a lot of different aspects. So we're telling a classifier that all these things that look very different should be called the same thing. And that again trips it confuses potentially the algorithm in terms of what is what. Another facet of this is that if we now look at copepods, for example, we have copepods from different classes, okay, with vastly different concentrations in the data set that actually look very similar, more similar to each other than the ones in the detritus class. And now we're telling the classifier that those are actually different classes, okay? So to say it differently, we have class imbalance. That's the term for the non-uniform distribution. We have, large variance intra-class and small variance between classes, and all that makes it a nightmare for many algorithms. So this is the result of a comparison between the measure plus classify approach and the deep learning approach that we did for many plankton imaging data sets. We won't look at everything, we'll just look at a subset of this. In green, you have the original approach. In blue, you have the convolutional neural network. And the first thing that we can look at is the accuracy on the top left here. So basically, how many images did the classifier get correctly? Okay, and what you can see that it's even for the old approach, it's actually quite high. The reason it's quite high in many cases is that it's a classifier that would classify everything in the dominant class would already be quite good. Okay, and that's actually red. The red bar here. So basically a really dumb classifier for UVP images that would just give the dominant class would be right more than 60% of the time. So of course, then when we try to be a bit more intelligent, we get to high numbers, but this is what we want to compare. And basically, in terms of accuracy, we see that CNNs bring a little to the table. Sometimes, in some data sets, it's actually quite a big jump, but in others, it's actually it looks like it's a really marginal progress. So that basically these new approaches don't bring anything to the table. Actually, then when we zoom on the classes that are specifically plankton, not the detritus typically, which is what you have at the bottom. So we have the precision and the recall, which are two metrics that you can gauge the quality of a classifier by. We see that for these rarer classes that have, again, small interclass variance, then we see a big jump in almost all metrics when we switch to deep learning. So this is what deep learning has really made a difference on. It's the ability to distinguish rare or difficult classes. So know that we have seen that basically for image classification to generate data, deep learning is really the way to go to get the rare classes. The question becomes which deep learning, okay? And a big question in deep learning algorithm is the size of the network. Basically, how deep, how big does it need to be, with the idea that the bigger the network, the more differences, the finer differences you can make. So bigger is better. So what we did on those same data sets that we started with a really simple network, which is called MobileNet because it can go on your mobile phone, that has 5.4 million parameters. So compared to a linear model with 2 parameters, of course, it's complex. For a deep learning network, it's actually not. It's actually in the smaller ones. And we get 90% accuracy, 91% precision, 92% recall, okay? So that's our baseline. And then we went, okay, let's try to do better by having a bigger model, by complexifying a bit the classifier part. We jumped to 7.5 million parameters, so increased by 50%, and no difference in accuracy, or any metric, basically. Then what we do is we change the convolutional part, the part that extracts the features, and we take a fairly reasonable network by today's standard with 25 million parameters and still no change. We want to go even bigger. Maybe we did not go as big. So in that case, we went to the biggest one. We go, an order of magnitude 200 million parameters and actually no change. So that means that basically the small network already did the job. So what we did then was go the other side, try to make it smaller, and we lose 1 million parameter compared to the first one, and again no change. And actually we even dropped the deep part for the last bits, and we see no change. So basically that tells us that there's a big gap between the measure and classify approach and the CNN ones. So this is the metrics for the measure and classify approach. These are the metrics for the different CNN that I showed, and this is the size of the model. Okay, so CNNs are bigger, but there's a big change in the quality of the prediction. But then between really big ones and smallish ones, it's almost the same. So basically for plankton classification, it seems that the shift to deep learning is really a big progress, but then which network exactly you use is not the main, but the images are such that it does not make a lot of difference. That is in the end to be expected, because we, as I showed you, many instruments take images that are a gray level, that are rather small, that have a uniform background. So in terms of features to extract or information to extract from those, there's not, it's understandable that a reasonably sized network is already enough. So I spent a lot of time on classification because this is probably the most where machine learning contributes the most to exploiting the big data that plankton imaging instruments produce. Okay, they produce a lot of data. Segmentation is oftentimes easy, and so it's really a classification problem. And then we get to normal looking ecological table that we can deal with. That said, with some instruments, segmentation is actually not that easy. So what you have on this slide at the top is the raw data taken by the ISIS instruments that I showed you initially, which is towed by a ship underwater. And this corresponds to 0.35 seconds of acquisition. And what we're interested in are all the small speckles that are here. And if we do just a simple, basic thresholding and take all the speckles that are bigger than a few pixels, then we end up with 2,500 objects to classify for every 0.35 seconds of acquisition, which is millions and millions of objects for a simple one-day deployment of over 6 hours and billions over a normal cruise. Okay, so that's just not, it gets to numbers that create problems even for classifiers, especially because of the dominance. It's an in situ instrument, so you have a strong dominance of a few classes, often classes that are not the target of the study. And so within a normal cruise, there would be 1.5 million objects, but only 1% of those would be planktonic organisms. So the classifier would have a really hard time finding those 1% with, we would have to have extremely limited error to then be able to trust the results. So one solution was to go towards what's called semantic segmentation, which is also something that deep learning networks such as the CNNs that I showed you can do. And you have an example of a semantic segmenter on the right, on an image of something that's not plankton. So the idea is that these networks, they would segment the object. So for example, the green here is identified as a bicycle. So the network has done the segmentation. So the identification that these pixels belong to a bicycle. It would also give a bounding box of this and an identification. So you do everything in one go. And the advantage of this is that it can use the full context of the picture to determine that this is the bicycle and this is actually the cyclist, etc., etc. So you can get to better identification by taking this context into account. You also see that even though this works quite well in that particular case, the mask is not very, very precise. So for plankton, we had to adapt this a bit. So it's just to mention that basically machine learning is also sometimes used for segmentation of planktonic images because the non-machine learning based approaches are not appropriate. And Finally, to go back to classification, what you've seen is that in the example that I showed, no classifier was at 100%. Okay, there's no perfect classifier. So what we in Villefranche developed in particular, together with colleagues, is what I'm showing you here, which is a web application where the images are stored with a lot of the metadata, including all the features that were extracted. And then, classification algorithm has already predicted them to be in a certain class. So we're looking at the potentially detritus class. So in that case, the classifier was really them, putting everything in one class. And we see there's copepods. So we click on a few and we put them in the, in the copepod box. Okay. So we have the identification of copepods. And then we'll go and pick another class. So we'll actually pick, these long, thin-looking things. So we pick a few examples, different size, different shapes, different orientations, and all those we put them in the fiber class. Okay, save. And so now we have a few examples. We'll do that with the ketognats again, different aspects, different size. Put them in a class. So there's, as you've seen on the left, there's auto-completion of the names. We have the full tree of life to classify stuff in. And then based on these examples, we can actually train a new classifier just for us based on, the few examples of copepod, ketognats, fibers, and detritus that we've classified. Okay. Here, what you have at the bottom are all the features, the classic features that were extracted from the image. And also we have deep features that are extracted by a deep network, the CNN that I explained earlier. And then we run a classification. So as you have seen, there's no coding involved. There's no need to be able to deal with Python or R code. Just, putting a few images in boxes and predicting them. What we see that if we go on the ketognat class, then the prediction is not very good. But then what I've done here is sort by score, meaning that the classifier will also give you confidence in its classification. And when we do this, we look at the high confidence image and we see that they're all right. So we can quickly validate, say, yes, this is ketognat. Yes, this is ketognat. And in a few seconds, we have 45 ketognat images classified this way. Same for the copepods, we can validate quite a few. Same for the fibers, we can validate quite a few. And then we can actually rerun a classifier to get a better prediction and go through the data quicker this way. When we do it this way, we see that operators that do this have a throughput of 2,000 to 10,000 images per hour, depending on the instrument, depending on the difficulty of the classification task. And that's 200 to 10,000 data points, ecological data points validated by a human per hour. So that gives us a tremendous amount of data to then analyze and extract information from, which is what I'm transitioning now. And I'll try to go quick on this because I'm running out of time. So once we have acquired this data, what do we do with it? So it's actually fair that I spent a lot of time on the data acquisition because this is where machine learning has made the most difference to deal with that big data. Because afterwards, actually, it's just normal ecological tables that we could deal with any usual technique. So, for example, sorry, this is a time series of plankton in the Bay of Villefranche, next to home, for me, and for different classes that were predicted by a model. And in that case, actually, you have 3 curves because we have the reality that was all validated by humans, and we have the prediction of a CNN and a merger and classifier approach. I won't look into the detail of these 3 curves, but what this shows you that we can quickly get to long time series with consistent images and consistent classification over the whole time series. And for example, for these Phaeodaria, which are unicellular organisms, we see that in the Bay of Villefranche, we see a clear trend in the abundance or in the concentration of these organisms, with almost none before the year 2000 or 1999, then some for about 10 years, and then higher peaks during spring for the next 10 years. So basically 3 shifts in the abundance of these particular unicellular organisms. So the first application is basically can give us a good longitudinal follow-up on an ecosystem. Now, if we switch to spatial, this is the data from the underwater vision profiler, which is, again, one of the in situ instruments that I presented earlier. And what you can see is that there's some a bit all over the world. And what we're looking at in the small circles is the proportion of the organisms that belong to the Rhizaria class. So the Phaeodaria were one kind of Rhizaria, so unicellular fragile organisms over all this. And the surprise here was the large proportion of Rhizaria in those in situ images, because this is not something that you see on nets, mostly because the nets will destroy basically these fragile organisms. If we sum everything in terms of biomass, in blue you have the copepods, in orange you have the Rhizaria, and the converse. In orange you have the copepods, in blue you have the Rhizaria, and what you can see is that near the tropics, the biomass of these two groups are actually quite similar, even though we thought previously that the copepods were dominating everything everywhere in terms of biomass in the plankton. Another application of this UVP data with a newer data set, so what you can see that we have a richer data set now, is that so we have 3,000, between 3,000 and 4,000 points all over the world, which is already quite good. We see differences in concentrations, but the problem, what we would really want is a map that looks like this with the interpolated concentration everywhere on Earth. Okay, that's of course is not possible from the data. We can't really interpolate the concentrations from there, because there's big gaps and the interpolation will not make sense. So what we do in that case is that we consider that the concentration of our organism of interest will be related to environmental variables, will be driven by environmental variables, basically. So we relate the concentration measured here to the temperature measured here, and we have 1 point. We do that for several points, and in the end, we have a curve of the response of the concentration to the temperature in that case. We don't do that with just the temperature, we do that with multiple variables. And to draw that curve, we actually use machine learning approaches. Many different are possible. In that case, we use gradient boosted trees. Once we have that model, it means that we can go anywhere on Earth where we know the temperature, and we have it from different sources everywhere at the surface in that case, okay? And then from the temperature and other variables that we used in the model, we can predict the potential abundance of those organisms. And in another place of the Earth, we can do the same, and then we have our complete map of the potential biomass of these organisms over the whole world. When we do this with many planktonic groups between 700 microns and a few millimeters, what we find is that this part of the plankton has a total biomass of 0.2 petagrams of carbon, okay, which is in line with other estimations. So this was satisfying, but in addition, we have the spatial distribution of this through that process. So we'll switch scale to go from very large to very small scale. Okay, what imaging instruments, in situ imaging instruments allow when you have really high frequencies to look at plankton concentration at high definition, and more importantly at the same definition that we can have on biogeochemical and physical variables. So this is a transect done with the ICS instrument that I mentioned earlier. This is 100-meter depth. This is 60 kilometers length. So we're looking at a mesoscale structure here. On the left, you have the coast. On the right, you have the offshore side, basically. And the white lines here are salinity isolines, as you can see here, that delimitate a coastal water mass from an offshore water mass, and with a front in the middle. And the black isolines correspond to the fluorescence, where we see a really strong deep chlorophyll maximum and stronger on the offshore side. So this is something that we used to see with glider data, with high frequency CTDs. We can get to these fluorescence, salinity. This is the oxygen. So this is something that we work with now in oceanography, and thanks to imaging and to the processing of the vast amounts of data that this generates. And then we can have a picture, in that case, of another kind of resalia, the Eolacanthidae, at the same resolution. And we can see that they accumulate where the chlorophyll is also accumulated. In the oxygen, we can see submesoscale recirculation with downwelling of water. And we can see that this affects these planktonic organisms in the same place. We can see an effect of the front, separating, some inshore from some offshore, dense parts, etc., etc. So we can really access to the biology at the same scale, or the biology of large planktonic organism, or large millimeter-sized planktonic organism, at the same rate and scale as the physical environment. So I'll probably skip that one, but what I want to highlight from this is that we can also access to life cycle parts that we had not seen before. So we know that shape of these organisms, we know that shape, and what we've seen on the images are intermediate parts that are specific distribution, specific ecology probably, that we can access directly in situ. And a last example that I want to show is that in images, there's not only information about the taxonomic identification of the organism, there's information about the shape and the morphology of the organism. So what we can do on the time series that I showed earlier, in addition of the identification on this time series, we can take the features that are extracted from deep network, for example, and then we can try to summarize the aspect of the plankton. This is done through a principal component analysis in that case, but it can be, other things. And what this highlights is that basically there's small organisms, larger organisms, there's dark organisms, there's lighter organisms, okay? And we have a quantitative description of these morphological characteristics. Now that we have that quantitative description, we can estimate the morphological diversity at any point in time. This is done by computing several indexes in that space. A simple one to understand is that if you have at a single date over the whole time series, if you have organisms from almost everywhere in that space, then it means you have a really diverse community. You have big ones, small ones, you have light ones, dark ones. If you have everybody in the middle, then you have a really, low diversity community. When we do this, what we observe is that in that particular time series in the Mediterranean, we have less and less zooplankton, so a significant downward trend in the concentration of zooplankton, but a significant upward trend in the morphological divergence, so basically in the one measure of diversity of that plankton. And that relates to the fact, to a basically well-known situation where we expect oligotrophic regions to be richer than eutrophic regions. This is known spatially. What we suspect is at play here is the same thing. We have the same, a single spot that is becoming more and more oligotrophic that supports a smaller and smaller concentration of zooplankton, but of plankton that is more and more diverse. And finally, the same approach, so exactly the same, except on a different data set. So we have, that morphological space, and then we can look at one aspect of that morphology in the environment. This is the Bay of Baffin, or Baffin's Bay, rather, northern Canada. And this was, 200 stations, roughly sampled during the ice breakup. On the left, you have ice. On the right, you have open water. And what we're looking at here are images of copepods that are on different parts of that morphological space, okay? So we're not looking for anything. We're just describing the morphology. And what we can see is that we have large ones on one end and small ones on the other end. The large one are associated often with a resting posture, which we see in a different, in a different parts of that morphological space. The small ones, they are in an active feeding posture. And so what we infer from this is basically we have large dormant adults. On this side, we have adults, but we also have, younger copepods that are actively feeding. So from the description of the morphologies, we're within one group, only copepods. We're not interested directly by the taxonomy, but from the morphology of individual organisms, we can go towards inferring behavior at the level of a community and several hundred kilometers. And that is only possible because we have a huge amount of data to start with that we can analyze through machine learning in an objective way. Okay, and with this, I thank you and welcome your questions.
Thanks so much, Jean-Olivier. That was really interesting to hear about all the different uses of machine learning in your work. So yeah, we'll go on to the Q&A session now. So please drop your questions for Jean-Olivier in the Q&A box, and then I will ask them. So I will start off with one of my own questions. So yeah, it seems that there's more and more use of machine learning in marine sciences in general, and as you showed, particularly for plankton ecology in the last couple of years. And yeah, you also mentioned that this competition for the computer scientists to get them more into working in plankton as well. So I remember from the big data document that we published, one of the recommendations was to stimulate more of these collaborations between the marine scientists and the computer scientists. So it would be interesting to hear more from you on that aspect. And what are other ways to collaborate with computer scientists from the marine science perspective, and how much, if there's scientists that want to use machine learning more in their work, then how would they go about that? If it's something new for them, and how much would they have to learn versus, collaborating with the computer scientists?
That's a very interesting and difficult question. I'll give my opinion, which is not necessarily the opinion of everyone. So I think first that it's easier and easier to get into machine learning because the tools are made easier and easier to use. So that means that to be, I would say, a consumer of machine learning, typically to be someone who uses machine learning to process data, there's, I would say, almost no need for collaboration. You need to, people with a biology training, such as myself, you need to learn, you need to, push yourselves towards doing this. There's some coding involved, necessarily, okay, but that's I think that's true for almost everything in ecology now, so that's not so much of a barrier. But so there's this to do, but then a lot of the tools are off the shelf, sorry. Even the deep learning things that I showed that were scary a few years ago are actually, I would say, even easier to use than other approaches now. Okay, now to move the needle, meaning that to not just be a consumer, this I don't think is possible by non-specialists. So currently, because of the state of training that is, separate in discipline, that implies collaboration. Okay, and the difficulty in that collaboration is that the problems that we face as ecologists are often, many of them are pure engineering. Okay, we have a bunch of data and we want to go through that bunch of data faster. And we see this as a computer science problem when it's not, it's a computer science problem, a computer science engineering problem. It's not a computer science research problem. So for me, a lot of these problems, if you cannot tackle them yourself because there's no off-the-shelf tool, I would say that the most promising way is to talk to companies, because there's more and more companies that do this, that have engineers who are happy and really satisfied to do these things. Particularly we see with ecological problems, that's something that some companies really have interest in helping with, which is great for us. And finally, if it's if it's possible to find a problem that is both an ecological problem and a computer science problem, then it's possible to have collaboration between labs in the two disciplines. Those are extremely difficult to find, and in my experience, take years to emerge. It's not, okay, I'll go talk to the computer guys in the next lab, present them the problem, and next year we have a paper out. That will never happen. And finally, I would say that to me, the next stage would be the solution for that is training, is to train people at the interface. There's a lot of it's a bit of a buzzword, but actually in some cases it works. There's a lot of training with majors and minors at universities, etc., and train people really at the interface between environmental sciences and computer sciences that might give rise to a generation of people that can actually tackle both. Currently, I don't think that's possible.
Yeah,
that's
thanks.
my long answer.
Yeah, it's good to know that there's a lot of the algorithms are available open source and off the shelf. So I have a question here from James Clark. He says, many thanks for a great presentation. For ecotaxa, you demonstrated a workflow in which each image was checked by a human operator. Do you believe it is still dangerous to quality control on just a subset of the data? And if not, what are the most important factors to consider when selecting a subset of the images for quality control?
Okay, thanks, James, for the question, which is also a very good question. So the goal of ecotaxa is indeed to help human operators to check everything that is on it, okay? That doesn't mean that every piece of plankton imagery has to make its way through ecotaxa, especially when we have instruments that generate hundreds of millions. Humans will never scale to validating hundreds of millions of images. It's not a computer science problem. It's a human in front of the computer problem. So we definitely do, in some occasions, only check a subset of the data. In that case, the most common, a well-known problem in image classification or classification in general is what's called dataset shift. Basically, the fact that the training set on which the algorithm is trained has an aspect that is different from one particular sample. So there are techniques to detect that beforehand, and basically so in the case of images, that would be we extract some morphological descriptors. We try to have a training set of images that are as representative of the diversity of real life as possible. Okay, that's the first step. Have a good training set. And then we can monitor, actually, if you have regular samples in space or in time, you can monitor whether that sample has a distribution in terms of morphology, not of class, because you haven't classified it yet, but of morphology, which has a distribution that is similar to the training set or different. I would say the most important ones to check are the ones that are the most different, because this is where the classifier is likely to make the most mistakes.
Yeah, and so we've got another question from Hee-Kyung Jung. So he says, regarding the non-uniform distribution spatially or within species, would there be any proactive measures to deal with that? For example, using the fixed data set for cross-validation? I'm
not sure what the non-uniform distribution refers to, whether it's a non-uniform distribution among the classes, or I don't know if you can precise your question. Is it the plot that I showed with the fact that there's a few classes with high abundance and other classes with low abundance, or something related? Okay, okay. There's no magic solution And so, I mean, one way to deal with this when trying different classifiers is to make it as even as possible. And that is possible by subsampling some classes. But actually, the non-uniform distribution is real. Okay, it is something that is in the real samples. So if you train a classifier with a uniform distribution and then apply it to a non-uniform one, then it will also make mistakes of a different kind, but it will make mistakes. It will actually overestimate the small classes. What we do is kind of a trade-off. I don't know if it's ideal. Well, first off, I know that this is an active topic of research in computer science. So there's theoretical papers talking about, non-uniform distributions. So the keywords to look for are, imbalance classification or class imbalance classification. So there's many different solutions. The one that we personally have experience with is to keep the non-uniform distribution, but assign weights to the different classes, saying that a mistake on a really common class does not cost a lot. A mistake on a rare class costs much more, so that we basically force the classifier to pay attention to the rare classes. And you have a knob to turn basically in the shape of those weights to put more or less weights on those rare classes to try to find some sort of compromise between representing well the real life, which is not uniform, and still having a classifier that detects the rare species in a good enough manner. It's a really hands-on and practical answer because I don't think it's a solved problem. I'm pretty sure it's not a solved problem yet in computer science.
Okay, we have another. Oh, thank you. He just says thank you. So I have another question. Because you mentioned that obviously there's the human component as well to classifying these images. And is there difficulty in finding people with the good expertise for this classification of the plankton? Because it's something that comes up fairly often in the marine board, that we discuss this lack of taxonomic expertise. So that's quite an important aspect in the process of this machine learning classification to get right. So do you have any thoughts about that, or what's your experience?
So yes, absolutely. The lack of taxonomic expertise, there's a lack of interest in this and sexiness of taxonomy, I would say. There was at the beginning, for plankton imaging in particular, at the beginning there was so at the beginning was 10, 15 years ago, this idea that the taxonomists would be useful to make the training sets, and then basically the classifier will do the rest, and we will not need the taxonomy. The taxonomists anymore. Our experience is that at the very least you need to check a subset. If it's humanly possible, you need to check everything, because in the end, again, if you're interested in rare species, if you're interested in low abundance or low concentrations in situ, if you're interested in small changes, you need the 100% accuracy or as close to this as you can get with humans. So in the end, we still need taxonomists, and there's fewer and fewer. Our limited and personal experience with this is that if we have a few reference people, it's possible to train people that are interested in this but have no experience in an acceptable time. But the reason is that we don't go as deep with images as we would with microscopic taxonomy. Typically, it's rare to go to species, etc. So the amount of information for one person to learn to sort correctly is smaller. So the problem becomes now that the traditional taxonomy textbooks, they are not really applicable in many cases. You're not looking at the same meristic characteristics on the images that you would under a microscope. So there's books to rewrite, which is also something that we're we have been trying to do for a while, basically publish together with the online tools to put images and have the computer sort them, have an online tool for humans to have a reference, have a guide on how to sort. So that doesn't, again, we absolutely need taxonomists, and it's taxonomists that will allow this also to go towards, finer classification.
Yeah, and so James Clark has a comment on that and another question. So he says humans are also imperfect at classification, and is that something that you've tried to take into account?
Yes, absolutely. So this is important to take into account when we look at the statistics of the classifiers. Okay, so I told you we get to 90% accuracy, let's say. A human, so you compare 90 to 100 and you're saying, okay, we're missing 10%. Okay, we're making 10% of mistakes. On the same task, a single human would probably be 96% or 98% accurate, but certainly not 100%. So the first thing is that, we should hold the automatic classification to the same standard as the previous data, which is not perfect. I would say that this, nonetheless, one big advantage of images is that several people can look at the same images and correct one another. So when a single human is fallible and will make mistakes, several people will probably not make the same mistakes and will never get to 100, but will be close enough to 100. So this is an exercise that we have done on subsets of some data sets. And the amount of mistakes that we see remaining in the data sets are on the order of, 1-2%, which is very, very good. Okay, yeah. Which is, and for this is of course very important for the training sets, because if you train with mistakes, then of course you can't expect the classifier to learn the mistakes. So on the training set, we make a particular effort to be as accurate as possible.
Yeah. Yeah, I think it's never possible to be 100% accurate. So, yeah.
We're humans.
So I've got a question from Paula Kellett, who's one of the science officers at the Marine Board. So she's asking if there's also autonomous ways to gather images and data about plankton at the moment, or is that sort of an emerging technology or something that still could be developed?
So it's emerging in the sense that, it exists, but it is currently small. Mm-hmm. So there's the underwater vision profiler, which I spoke about. There was a the fifth version, which is a 1.2 meters high, and fairly bulky. No, not that much, but basically it's quite big. But the new version is smaller and can go onto autonomous floats, for example, or gliders or ROVs, so partially autonomous, let's say, or AUVs. And that has happened in the sense that a few floats have been deployed with that imager. A couple of gliders also have been deployed with that imager. For gliders, AUVs, ROVs, the situation is actually not very different from a technological point of view. It's not very different, because then we get the data, we get the images, we can sort them the same way as before. Okay. For the instruments, it's very different because you need to be super energy efficient, etc., etc. But for the data flow afterwards, it's fairly similar. For floats, then it's completely different stories, because Argo type floats are not recovered, meaning that the images stay on the instrument. There's absolutely not enough bandwidth, and it's not close to happening that we can send the images via satellite communication back on land. That will not happen in, I don't want to give a number of years, but it's several years. Okay, that means that the classification has to be done on the instrument, directly on the float. So we have been working on this for several years now, and the first instrument with embedded classification have been released. What we see is that we have to be extremely energy efficient for the classification too. That rules out completely using convolutional network. And so we fall back on the the type of performance that we had before. So basically what we see that we're losing 10% compared to the state of the art. So basically we have a bit noisier data, there's more mistakes, but on the other hand, we have a lot of data that we would never have been able to collect from a ship, because you can have several floats in the water at the same time. Having several ships is not possible. So it's in infancy. I think we'll see the benefit of this once enough have been released and we can aggregate data at a level where the error cancel out, basically, and we can see big patterns. Yeah. That's a few years, couple of years.
Yeah. Yeah, I'm interested to see where the field will evolve in the coming years. And I think particularly there's been a lot of interest in digital twin oceans. So I think machine learning will play a big role in that. So yeah, it'll be an interesting field to keep an eye on. So yeah, we'll have to wrap it up here, but thanks a lot for your time, Jean-Olivier. It's really nice to chat to you this afternoon. And I have one more slide to announce our next webinar. So it will be on the 19th of January. And the speaker will be Dr. Chris Freemantle. He will be discussing multidisciplinary understandings of indeterminacy, uncertainty, and risk. So he actually comes from the arts field, and he's on the committee for our Embracing the Ocean program. So he'll be making comparisons between uncertainty in arts and policy and science. So it will for sure be an interesting discussion. So thank you everyone for attending, and yeah, see you next time.